of life, in what form is it? Chromatin. For a cell divide, materials got condensed like copies. Once it becomes visible, what do you call it? Chromosome. Which is true.
B.
Chromatin is made of DNA. Chromatin is the bigger entity. DNA is a part of it. You wrap them in proteins, and it looks like a bead, a string of beads. The proteins are called histones, and the actual beads are called nucleosomes. The DNA strands composed of sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. So B's and D or D and B. The red ones are, I didn't mean to, but the red ones are correct because G1S, G2M, or if you put the M over here, you still got the same order. There's no way that mitosis goes straight to synthesis. And no way does synthesis go to G1. The two blue ones it's not possible. And G2 didn't go, but that's fine, but this won't happen. On this one, this will happen, but this won't happen. And that won't happen either. So, of course, on the paper test, there ain't no reds and blues, it's all just black and white ink. All right? What's G stand for?
Gap.
It stands for gap. Too many folks think it means for growth. Now, I know it's growing, but it stands for gap. It's the gap between mitosis and the next synthesis. Mitosis and the next synthesis. That's the gap. Okay. Well, which letter do you find cytokinesis happening?
G2.
It's happening at B. When this crosses over, you have the two cells produced, so right there's where it happens. I'm going to do it right now and take these letters off so it never get confusing.
I didn't do it.
Too much. What is the first stage?
G1.
It's G1. I got to do it here too. And the only part of interphase where nothing is happening except the cell is growing is G1. G1. Because in S you're making more DNA, and in G2 you're making more organelles. When you find DNA being replicated, it's called synthesis. It's the S phase. I'll go back big now. Okay. What are you looking at there?
Two nuclei and one nucleus.
Yeah. Here's the cell. That's a nucleus with two nuclei inside it. So the answer is this one right here. This is a nucleus. You see the cell right there. And this nucleus has one nuclei. Some have three nuclei. There's no rule that says only have one. All right. Now in G1 phase, how many chromosomes do you have? Just the one. This is what you have in G1. And then it doubles itself. So in G2, in S phase, what do you have? Right here. Now it's two. Trick question comes up now. In G2, what do you have? Still CD. Because this happens during mitosis. You're not even in G2. Follow me? So in G2, this is still present like it was in S. You won't have this happen until you leave G2 and enter mitosis. So some folks may have put G because you were thinking I'm doing A. I wasn't. So CD happens twice. It happens in the it happens first in synthesis, and it stays that way in G2, and then in mitosis, they split. So you shouldn't have said G at all. This one. The one word that you find organelles being replicated, that's G2. It's got to do that just before it divides. Cancer is a disease, and that mass of cells is called a what?
Tumor.
Tumor. You sound like Arnold Schwarzenegger.
Spell tumor. T.
She does that. Yeah, that's what Arnold did. What show was that? Was that the kindergarten cop? Yeah. Now, when I say tumor, I think of Arnold. And now I think of Hugo saying.
It was real heavy.
You guys
22.
only talking about my accent.
If you don't have this, if you don't have it,
Get
what's
sick
gonna
of
happen?
it. No, you, the team.
It won't
Hey,
go
I'm not the one that brought it up.
Okay.
What's that?
You guys get a life and leave me alone. I was talking about my
She
accent.
might beat you up.
I didn't bring it up.
Because she talked about her husband already.
We all have an accent in here.
No, don't talk about me. I have an accent. I'm
You got an accent.
I
You
do.
sound different.
Yeah, everybody sounds different.
We all sound different.
Yeah.
That's not being critical either. It's just, you sound different. All right? Well, if you don't get CDK, and that makes, that, so it's C, it's B again. Because these two go hand in hand. Without them, it just grinds to a halt. Well, that's why you don't run away with cell division. This is fighting cancer. Okay? Well, which two of those can lead you right into cancer? The first two, A and B, the two protein enzymes. What's it really? It's just a nucleus. It's not a cell. It's just a nucleus dividing. And remember PMAT? Did you forget the word? Prophase, metaphase, anaphase, telophase. PMAT. The process of splits, that's cytokinesis. That's when the cell, nucleus divides first, cell divides second. During this phase, the chromatin becomes visible. That's where I just stopped myself reading, because that's prophase. And I could keep going, but when you see chromatin become chromosomes, that's prophase. That really is what? Is it one chromosome or is it two chromosomes? It's really one that's replicated. So this is all one chromosome, but she's got two halves called chromatids. Now when they separate, you're right back to having two chromosomes. But that right there is one chromosome that's made a copy of itself, and now I got two of me. Okay? The centomere is A, the kinetochore is C. So this is the glue that holds them together. And this is the attaching point for the spindle coming in over here, that centrosome, the diaster. It's going to pull apart one day. If they're down the middle, that's going to be one thing. That's metaphase. And when they're being pulled apart, they take on that V shape. That's anaphase. The chromosomes are as far as they can get, but the key word is cytokinesis is starting. That's telophase. It won't be anaphase because in anaphase, they're not all the way there yet. They're still moving. And I know cytokinesis does start in anaphase too. But to make the question come up to the telophase, I had to tell you they're at the far ends. They're as far as they can go. That's telophase. Which is prophase?
See.
Yeah, this is interphase because it's still inside the nucleus. When the membrane goes away, these things spread out and get a little lighter colored. Here they're real thick. So now the membrane's gone, and I start to see individual masses. So there's prophase. And metaphase, only one of them down the middle is this one here. That's prophase. Anaphase, they're being pulled apart. Do I see any V's at all? There might be a V right there, baby. But obviously, nothing else fits to be separated but that one. That's anaphase. And telophase, of course, right here is telophase. I'm eating it up. I got my two new cells. Don't look like they're really divided yet. They're still in the process. And then cytokinesis, of course, is D again. I looked here carefully. I don't see any kind of dipping in. Also, I didn't show the whole cell either. If I got more cell, I might see some dipping in, but I did on purpose. I didn't want to show you a conflict between these two. So that's the only one right there that shows the pinching in the cytokinesis. Interphase of that, of course. Now the other one, I got these two to look at. If I can see the chromosomes, I'm in prophase. If I cannot see them, I'm in interphase. So there's prophase. Down the middle, anaphase. I don't know if I see a V. I see a V there. I see a V there, but truly they're being pulled apart. That's anaphase. And then telophase is the last one, and it looks like they're pretty much they'll soon be separate. And of course, that also shows the cytokinesis, so it'd be E and E back to back. Anaphase back there. I mean, I have a nice, strong nuclear membrane. Here it's gone. So interphase becomes prophase when two things have happened. The nuclear membrane is dissolved away, and this chromatin has gotten thicker, and I can now see chromosomes. Okay. These two are usually the ones that folks get backwards. But after if I tell you, I don't know how you would, because I can see here. I see daylight in there. That means chromosome. Here it's so thick. That means chromatin. So that's it. All right. Let's go back and do some. I do want to save that.
After doing that, I've decided that I would rather take the big test on Thursday and the the PowerPoint test on Wednesday.
We done decided that.
Huh?
We done decided that. There was no reason to take that paper test when I said the PowerPoint. Because the PowerPoint becomes a paper test. And we decided we're doing the PowerPoint
On Wednesday.
and doing the paper test on Thursday. So for that very reason. So you can see you got a warning shot.
Yeah.
There's no reason to do it the other way around.
Okay.
I missed a lot of this stuff.
All right.
I got all the slides though.